Hello and welcome to Chapter 2 in Biopsychology. In Chapter 1, we reviewed the basic premise of biopsychology, of the biological explanations for everything that we encounter in the world of psychology, and some basic factoids about biopsychology. Now we're going to start getting into some of the building blocks that build up what we know about the human nervous system, and we're going to be starting here in Chapter 2 with nerve cells and nerve impulses. So let's just get right started into this. So, first of all, within the nervous system, there are essentially two types of cells. We have neurons and we have glia cells. Now, neurons, which you're probably more familiar with, are communication cells. These are basically cells whose primary function is to communicate information from one part of the body to the other. Some of those neurons receive information, something like sensory neurons receive information from the outside world. Others transmit information, like the next neuron down the line. You have the one that's in the retina picking up on light, and then the next neuron down essentially has the responsibility to continue transmitting that information, eventually getting it all the way to the occipital lobe for processing. Different neurons specialize in order to be able to sense different kind of information and in order to send different kind of information, all basically based on their function. Now, the glial cells, or glia cells, are cells that are also associated with the nervous system. Their primary role is to provide a support structure, because otherwise the neurons are loosely held together, and the support structure from the glia help them to stay close to one another. They transport nutrients in order to feed the neurons. They are involved in waste management, although not in any way associated with the sopranos. And they regulate blood flow. Now, in the nervous system, there are about 86 billion neurons and 86 billion glia cells. So about half the nervous system is based on neurons and half is based on glia. Now let's take a closer look at the neuron, the physical structure of the neuron. This is my classic graphic that I use in all of my classes for just going over the anatomy of a neuron. So, like all cells in our body, there is a cell body or a soma, and in that cell body there is a nucleus. The nucleus, of course, contains all the genetic information that is contained in all of the cells in our body, but this particular one has been specialized in order to create a neuron. Most obvious, we see dendrites branching out from the neuron at both sides, all over the neuron. Depending on the structure and type of neuron is, you might see dendrites all over the place, or just might see a neuron that looks very much like this image. There is one part of the cell that we identify as the axon, and it tends to be kind of a longer dendrite. When you look at actual slides of neurons, it's difficult to really determine which part of the neuron is the axon. Axons, however, have the distinct quality of having Schwann cells, which we will talk about in glia cells, because Schwann cells are a type of glia cells that produce a fatty substance around the axon called myelin. Myelin, which we will learn later, increases the efficiency and speed of our nervous system. In between the packets of myelin, we find ourselves nodes of Ranvier, and these are openings along the axon where ion channels can open and boost the neurological signal as it goes on down the line. Finally, we have the axon terminals, which are the synapse areas that we're going to be talking actually in the next chapter. In Chapter 3, we'll be looking specifically at the synapse and the kind of magic that happens there. So that's the neuron in general. Glia cells come in a number of different types. And the first one we're going to talk about is astrocytes. Now, astrocytes, very important part of the support system for neurons. They support the biochemistry of endothelial cells that actually comprise and manage the blood-brain barrier. We're going to be talking about that a little bit here. The astrocytes keep those cells in place so that the blood-brain barrier can maintain its function of being a barrier to larger molecules such as viruses and other sorts of invading organisms. So it's very, very important because we're going to see that the brain needs to have a specialized set of defenses against infection. Astrocytes also maintain the extracellular ion balance that we're going to talk about when we talk about resting and action potential, and I'll mention this again. The regulation of blood flow, and finally, the repair and scarring process of the brain and spinal cord. Now, when I was raised in psychology, I was told the nervous system doesn't repair itself. Now, it does in a way, and certainly massive injuries, such as spinal cord injuries and whatnot, are really, it's pretty rare that we get any neurological growth to repair that at this point in our understanding. However, there is some repair of the area in which the injury or the cut happened in the nervous system, and there's a buildup of scar tissue in order to build up the support for the surrounding neurons to continue that function. That building up while supporting the neurons in the area also inhibits any sort of possibility of growth of neurons. So it's sort of a plus and minus aspect of the actual function of astrocytes. And that's kind of a point of research going on right now about how can we modify the actual actions of astrocytes in order to allow for the development of new neurological tissue. So something that's currently in the research. Now, another type of glial cells is microglia cells. And as you heard me mention before, the brain needs special forces or special resources in order to fight off infection. Microglia are the first and foremost immune defense in the central nervous system. They react very quickly to reduce inflammation. Now, inflammation is the body's natural process of fighting off disease. So, more blood flow to that area, it brings pain-reducing chemicals, it brings more oxygen, it brings all the nutrients that area needs to do the repair. But the negative side effect of that is you have swelling, pain, and sometimes that process can be out of control and inhibit healing. So when we have inflammation in our leg, we may take some anti-inflammatory in order to reduce the pain and enable us to function while the body continues to do its repair. The brain, for one, doesn't experience pain, not directly. There's no pain neurons in our brain. When we have a headache, that's actually the pain neurons that exist within our meningeal layers, and there's actually swelling of the blood vessels associated with that typical migraine headache. Now, we don't need the pain-reducing parts. However, remember what I said about the structure of neurons. Neurons, unlike some of the other tissues in our body, are not really connected to each other. They come very close, and those synapses are almost touching, but they really don't. So the glial cells and the other support mechanisms are holding them in place. Any sort of infusion of blood or liquids or fluids or anything into that area and expanding that can cause significant damage to those areas. So when we think of inflammation of the brain, that's a really bad idea. We won't feel it, but there can be significant damage to the tissue there. So what we see is those microglial cells reacting very quickly in order to attack any invading organism and therefore avoid the whole inflammation game altogether. So that's a very, very vital aspect of microglial cells. In addition, we understand that most antibodies are too large to pass the blood-brain barrier. Therefore, the microglia represent a specialized force, very different than the other disease and infection-fighting parts of our body. This is specifically to the central nervous system. And finally, they are a scavenger of the brain for plaques. Now, plaques are areas of the brain that are they're sort of scaly and clogged together, and bunches of dead cells and whatnots, and the microglia cells clear those up. Now, plaques, or neurotic plaques as they're known, are a common symptom associated with Alzheimer's. We're not sure about the cause-effect relationship there, but we know that individuals with Alzheimer's, when we've looked at their brains after they passed away, that we find these neurotic plaques. So there might be a relationship between the function of microglial cells and its ability to clear up plaques and the incidence of Alzheimer's. Now, that would be information that we would use both to prevent Alzheimer's and also to treat existing Alzheimer's patients. Now, moving to our next type of glial cells, we have two types here that essentially perform the same function. We have oligodendrocytes. I'm glad I got that on the first try, so I don't have to say it again. And we have Schwann cells. Like I had mentioned Schwann cells when I was looking at the neuron itself as the part of the glial cells that produced myelin. Both of these types of glial cells produce myelin. This one, I'm not going to attempt that word again, is a buildup of myelin in the central nervous system, the CNS, the brain and the spinal cord. This is the production of white matter in the brain and spinal cord. Schwann cells produce white matter in the peripheral nervous system. So both of these types of glial cells do the same function. Now the final one we want to look at is radioglial cells. Now these are vitally important specifically during neuron migration in embryonic development. If you've had the chance to take developmental psych, you have probably come across the notion that in the developing brain and brain tissue, there are a number of processes that occur. And there's neuron proliferation, that's up to 250,000 neurons per minute. There is the neuron dendrification, synaptogenesis, synaptic pruning, and then there's this very curious one, neuron migration. Now that's when neurons that are not quite specialized crawl along these glial fibers in order to find the place where they're eventually going to settle down, grow up, get a job, raise a family. Kind of think of it that way. You have the progenitor cells at the base producing these neurons that then travel away from home and go and settle down for their new job and change once they get there. So to help with that imagery, I have this graphic here. We'll take a closer look at it. Here we have the developing brain. And to point out some of the structures here, this is a little bit along the way. This is not early developing, but we have these ventricles. These are spinal fluid filled areas of the brain that we have throughout our lifespan. And then we have this this bottom layer that we're going to call the ventricular zone. Now these are where these progenitor cells are producing new neuron tissue. Then we have the white matter. And then on the outside, that sort of characteristic convoluted part of the brain that we recognize as a brain is the neocortex here, soon to become after birth and then throughout our life, the cortex. Now, if we take a look at this sort of cross section so that we can see what's happening between the ventricular zone and the developing cortical plate, and we blow it up, here we have the ventricular zone, the progenitor cells producing new neurons, and then the glial cells creating glial fibers that exist and provide a way in which these specific new neurons can crawl up and travel to the part of the brain where they're going to reside, up here in the developing cortical plate. Now, the mechanisms that undermine, that are under this process of migration are still very, very poorly understood. What starts the migration, what ultimately stops them, the biochemical atmosphere, location, 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 right? So the neuron children move out, and then they find a place where they can be who they want to be. And let's say it's in the back of the brain, and that neuron develops into the capacity to process visual information. Now, that's different than the other. Maybe its brother goes off and comes up here into the motor cortex, and they're working on developing, a sports ability and be able to dribble a basketball, talking to its cousins back here in the cerebellum, which is very important in coordination and whatnot. So we see these neurons traveling along. It's kind of really freaky and amazing to think that these neurons actually sort of reach out and grab, and they're sort of crawling along these glial fibers. And that covers the last of the glial cells that we're talking about at this point in the course. Now, I'm going to transition to another topic, and that is the direction of neuron transmission, and kind of get this on the table so that we understand this as we approach taking on the notion of action potential, the actual biochemical process of the neuron firing. So I have a graphic for this as well, and here we have this graphic. Here we have the spinal cord, and we have nerves that are in, like, say, pain cells on the skin that are coming back to the spinal cord, and then muscle tissue and the neurons connected to the muscle tissue that's coming. You can almost think of this maybe as a reflex. Let's say this is the skin on my hand. Now I touched something hot with my hand, and that information starts from the skin and travels to the spinal cord. And the nerves that take that information from my skin to the central nervous system, in this case the spinal cord, are called afferent nerves. On the other hand, well, that's probably poor terminology. Let's just call it the same hand. My spinal cord gets really quick on this and says, pull your hand away, and sends information from the spinal cord out to my muscles in my arm to pull my arm away from the hot surface. Now, when the neurons are moving in a direction away from the central nervous system, we call those efferent nerves. Now, the point of this is these are different. They're not two-way. It's not like the pain neuron sends another message to the central nervous system and then the message is sent back. That pain neuron is traveling in one direction to the central nervous system. It's a whole other set of processes, a whole other set of nerves that send information back to, let's say, the muscles in that area, the muscles in my arm. Now, when we look at a neuron actually involved in a neuron impulse, we are always mindful that it's moving in a direction, whether it's one nerve and we're actually watching the development of the action potential in the cell body, and then it produces enough electrical charge to signal the actual discharge of that electrical activity, and then it travels down the axon and then to the terminal buttons. Or if we're looking at a series of neurons, we always have this sort of sequence, and it's in one direction. So when we get into the synapse, we're going to have the presynaptic neuron terminal button, and we're going to have the postsynaptic terminal button, and information only goes in one direction. whether it's toward the central nervous system or away. It's always in that one direction. So let's take a look at nerve impulses now. And the first I want to talk about is the resting potential. Now, this is the state of our nervous system when it is not activated. So let's get our graphic here. Here you can see the cell is portrayed in this white circle. This is the actual neuron, and you can see the mechanisms that are in there. You see the sodium-potassium pump, and then you see the extracellular fluid. Now, we're going to be talking about electrical charges, positive charges, negative charges, just like in batteries. In fact, this mechanism that we're talking about right now, if you're familiar with the movie Matrix, Matrix portrays this science fiction story of aliens taking over the earth, and that our real physical reality is that we are being raised in these pods and used as batteries. And the actual scientific concept of that is we actually do produce electrical charge in our body. Our nervous system does it. We're about to look at that mechanism right now. And that's kind of where they got that idea that aliens could use us as a little Duracell. And so there's not a whole lot of electricity going on, but if you had billions of them, I guess you could probably, charge your laptop or something. So here we have this mechanism here. This is the resting potential. There are two ions principally involved in the development and the propagation of neuron signals. The first one is sodium ions. These are positively charged sodium ions. The next one is potassium, equally positively charged. And so it is the relative degree of positive charged ions on the outside or on the inside that create the electrical charge along the boundary between the cell and the extracellular material on that membrane. In resting potential, their sodium channels are closed. This means there's no sodium necessarily traveling. The openings that are created, that we'll talk about in just a moment, that allow sodium to come into the body, come into the cell, are closed at this point. Okay? Another mechanism that is maintaining the status quo of resting potential is this sodium-potassium pump. Now, the sodium-potassium pump is a mechanism that provides for a balance between the external and internal presence of sodium and potassium. And the way it works at the resting potential is it is continually pumping out sodium and continually pumping in potassium, but it does so at a 3-to-1 ratio. So for every 3 sodium ions that are pushed out of the cell, only 1 potassium ion is actually brought into the cell. Over time, this creates largely an absence of positively charged ions inside the cell, so a negative charge there, and an excess of positively charged ions on the outside of the cell, so you have a positive charge. And here we have the resting battery. Essentially, the negative charge is what travels. So here we have this resting potential. The cell is negatively charged in an environment that's largely positively charged, and there's no electrons moving at that point. That's the way electricity operates. Now, if I was going to go back to my skin sensation and I put my hand on a hot stove, that stimulus on pain neurons actually is the mechanism by which those sodium ion channels are opened. So when I do something like that, or something bumps into me, or I feel like a soft fur of the cat, or I hear something, whatever the external stimulus is, the stimulus first and foremost in those specialized sensory neurons actually opens up the ion channels. And we start moving into the process of not resting potential, but action potential. So something comes along, either there a temperature, or I hear something, or I see something, feel something, taste something. Something happens and the ion channels open up. That means that those sodium ions that have been being pumped out at a 3-to-1 ratio to the potassium, they start moving into the cell through these channels. And we'll be watching a movie about that in just a second. As the positive charge begins to accumulate inside the cell, the relative negative charge starts to exist on the outside of the cell. This is a cell that is building up that energy in order to fire. Now this energy builds up, the negative charge builds up on the outside of the cell, it reaches a particular threshold, and then the neuron fires. So this is the mechanism as we understand it, the way the sodium and potassium are involved in sort of creating either a positive or negative charge along the membrane of the cell. Now to help with this, I'm going to play a couple movies. The first movie I'm going to play is going to be, and these are all in the course book, is going to talk about what I've already said. It's going to go into some detail about how the ion channels open up, but then it adds the really cool piece about how they get closed again, and the direction of that neuron impulse happens by the synchronization of opening and closing of ion channels. And then the next video that we're going to go into is actually going to examine a little bit more closely the effect of the myelin on the axons in order to increase the efficiency, speed, accuracy of the nervous system there. So let's start off with this video about action potential.
A nerve impulse is an electrical current that travels along dendrites or axons due to ions moving through voltage-gated channels in the neuron's plasma membrane. Voltage-gated channels open and close in response to an electrical voltage, so they are affected by changes in electrical charge around them. When a neuron is at rest, a charge difference is maintained between the inside and outside of the cell. This charge difference is produced and maintained largely by active transport using sodium-potassium pumps. The pumps send sodium ions out of the cell and bring potassium ions in. While other channels allow some flow of potassium ions back out of the cell, the sodium ions cannot easily get back in to replace the lost positive charges. The overall result is that the exterior of the cell has a net positive charge and the interior has a net negative charge. The difference in charge between the interior and exterior of the cell is called the resting membrane potential. A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions flow into the cell, lessening the charge difference at that location. If the change is enough, it will cause nearby voltage-gated sodium channels to open. This allows so many sodium ions to flood into the cell at that location that the membrane there is depolarized, with the local region inside the cell having a net positive charge and the outside of the cell having a net negative charge. This affects neighboring voltage-gated sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to restore the resting membrane potential. The voltage-gated sodium channels close and voltage-gated potassium channels open. This allows a rapid flow of potassium ions out of the cell, repolarizing the membrane so that the inside is again negative and the outside positive. This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential and to reestablish proper concentrations of sodium and potassium ions inside and outside the cell.
It's such a great video. I really like the animation and how it really shows that progression along a particular direction. So you have those sodium channels opening up, creating the, offsetting with the resting potential and creating that negative charge. And then you have the release of potassium, positively charged ions, back into the intracellular space to return that part of the cell to its natural resting potential. Now this particular video, the next video is looking at the same process, but taking into consideration the effect that myelin has on, in this case, the axon on the neuron.
Saltatory conduction. Conduction through myelinated nerve fiber. Due to the myelination of neurons, action potentials may only occur at the nodes of Ranvier. Myelin is made up of insulating cells, which means depolarization cannot occur in myelinated regions. Between these cells, however, there are gaps known as nodes of Ranvier which are unmyelinated. As depolarization cannot occur at the cells making up the myelin sheath, the wave of depolarization can only occur at the nodes of Ranvier. Thus, action potentials appear to jump from node to node when traveling down an axon, and this phenomenon is known as saltatory conduction, and it serves as a means of increasing the rate of propagation of an action potential. Not only does saltatory conduction increase the speed of impulse transmission by causing the depolarization process to jump from one node to the next, it also conserves energy for the axon as depolarization only occurs at the nodes and not along the whole length of the nerve fiber as in unmyelinated fibers. This causes up to 100 times less movement of ions that would otherwise be necessary, therefore conserving the energy required to re-establish the sodium and potassium concentration differences across the membranes following a series of action potentials being propagated along the fiber.
All right. So that is our video presentations for today. That's it for this particular chapter. And so let's take a look at the assessments associated with this chapter. And first, we have a discussion about glial cells. And so I keep on adding that L because that's how I learned about glia cells, glial cells. Either way, having a discussion about glia cells and their function, and then a 2-question quiz. One of the questions is about heroin versus morphine, and this has a lot to do with doing some research based on the size of these molecules and how they're able to get past the blood-brain barrier. And then a question about essentially potassium blockers, something that keeps potassium from moving. So if we consider the process that potassium has, the role, that potassium has in terms of the action potential, what if that was blocked? What would happen? If the potassium can't get out, what happens? That's what I'm looking for. I'm not looking for the physiological effect or how would I feel if that was going on. That's not it. We're looking at the biochemistry here. What would actually happen if something was blocking the potassium channels so that the potassium couldn't get out? Do that sort of thinking about, because we prescribe medicines, and we're going to be getting into a little bit of that when we talk about neurotransmitters. We prescribe medications that impact the sodium channels, the potassium channels, in a way where we want to get a therapeutic effect in that part of the brain. So we'll be taking a look at that in the next chapter. So this is chapter 2, and Hope you enjoyed it, and I will see you for Chapter 3.